probably one of the longest, I think, from Brazil.
Yeah.
So we welcome you here in Basel, Switzerland. So your next presentation will cover in vivo and in vitro evaluation of polymer particles and liposomes on myocardium targeted deliveries. So it's all yours.
Thank you, Chair, for the introduction. First, I would like to thank the organizer for inviting me to CLINAM. It's a great pleasure to be here. The cardiovascular disease is the leading cause of morbidity and mortality worldwide, with the number of fatalities projected to increase to more than 24 million from 2030. And particularly in the case of myocardial infarction, despite of advances and benefits of current therapies, these approaches do not promote tissue regeneration. And emerging therapies like stem cells, biomaterials, gene therapies, or even the administration of growth factor represent great hope for cardiac repair. But the myocardial delivery is a very important critical point. So, for example, the use of growth factor was evaluated by several clinical trials. However, the results from the 3 larger clinical trials were disappointing. It has stimulated a resorption and carrier system for delivery of biopharmaceuticals into the myocardium, because in the case of recombinant proteins, we have a short in vivo half-life, the problem of the protein instability in vivo, and undesired vascularization non-target sites in the case of angiogenic peptides. So here, this is the kind of nanocarriers for delivery of biopharmaceutics in the myocardium, like nanofibrils, hydrogels, liposomes, and nano and microparticles. We have demonstrated the efficacy of PLGA microparticles on the myocardium. In the myocardium, here we can see PLGA microparticles prepared by solvent evaporation method and injected, or in fact, the myocardium using a rat model of ischemia reperfusion. And in this video, sorry. I don't know how can, well, here we have a video, but I can't to play that. This is a video showing the intramyocardial injection of the microparticle suspension on the epicardial surface. So, yes, this is the video here. Well, this is the intramyocardial administration of microparticle suspension on surface of myocardium. Here we can see the suspension entering in the surface with a very good deposition of the tissue. So, after this administration, these kind of particles were able to remain on myocardium for 30 days, 60 days, and until 92 days. Also, this is VEGF-loaded PLGA microparticles with a very good in vitro and in vivo biologic effects, like endothelial cell proliferation and receptor activation. In vivo, these particles promoted neovascularization and a positive remodeling on tissue, mainly in the left ventricle wall. So we evaluated the feasibility of PLGA microparticles to deliver other growth factors like FGF1 and neuregulin-1 this approach was able to induce positive benefits in a rat model of myocardial infarction. In vitro, we can see control release properties of these microparticles and stimulation of endothelial cells. Next, we can see the in vivo benefits of this kind of formulation using the combination of two recombinant factors in a chronic heart model of infarction. Here we can see the positive, Here we can see the positive remodeling of the tissue, no fibrosis, neovascularization effects, and the recruitment of stem cells to the site of tissue injury. Next, moving to liposome research, we can see the potential, because we are evaluating the potential of liposomes for myocardial target to deliver of biopharmaceutics. A few reports have been published on literature in the last years. We can see some reports using liposomes to deliver biopharmaceutics in the myocardium. Well, we prepared liposomes using the lipid film hydration, and next we performed the liposome characterization by particle size at a potential and transmission electronic microscopy. So we have performed in vitro assays to evaluate the cell biocompatibility of these liposomes using fibroblast and cardiomyocyte cell lines. Here we can see that these liposomes were compatible with these two cell lines. And next, we have performed in vivo experiment using a pharmacological model of myocardial injury, that is the isoproterenol-induced myocardial infarction in mice, and to evaluate liposome deposition and liposome tissue distribution. So here we can see the procedure. This is the experimental setup. Here you can see an animal platform with a transducer. This is a transducer, and here the rat on the platform under anesthesia. And here a video showing this procedure. Yeah, here we can see by using two-dimensional echocardiography. Here we can see the needle reaching the left ventricle wall for delivery of liposome content. Well, prior to liposome injection, we performed a validation using administration of even blue dye. This was a very important validation, because here we can see the injection point. Here we can see the injection point on the left ventricle wall, and the tissue distribution of the dye content towards left ventricle and aorta. Next, this is the histopathological evaluation of this approach, where we can see that large liposomes induced inflammation in both myocardium and epicardium sites. In contrast, the small liposomes induced inflammation in myocardial sites. Next, we performed heart retention of rhodamine-labeled liposomes. This is fluorescent liposomes of several sizes, and in this approach, we could confirm the liposome deposition of myocardium for 3 days, and in the case, and here we can see a more intense signal of heart tissue injected with 479 size liposomes. Next, And finally, this is a tissue distribution study of these liposomes, where we can see no fluorescent signal on other organs like kidneys, lungs, liver, and spleen. That's indicating no migration of these liposomes towards solid organs. And finally, no changes on tissue structure and organ microscopy, as we can see. So, finally, as conclusions and future direction, PLGA microparticles are efficient systems to deliver growth factor in animal models of myocardial infarction with biocompatible and using a biocompatible and FDA-approved polymer with a long-term retention in heart tissue using an animal model of myocardial infarction. But it's necessary to develop non-invasive delivery method. Moving to liposome research, we demonstrated the in vitro and in vivo biocompatibility of these nanocarriers by ultrasound-guided transthoracic injection, but it's necessary to determine long-term retention on myocardium, and finally, to encapsulate biopharmaceutics for delivery in heart tissue, because this is this is a great hope for cardiac repair using this kind of biopharmaceuticals. So, thank you very much for your attention.
Thank you very much, Dr. Formiga. Question to his presentation? No heart specialist here? Oh, okay, good.
Thank you for your lecture. I would like to ask if you try to give the PLGA nanoparticles IV. I mean, not directly to the myocardium.
At this moment, this approach is ongoing. We have no results yet, but this is the very good objective, because this is a non-invasive delivery method.
Major obstacle for the nanoparticles.
Yeah.
Anybody else? Okay. Everybody understood everything what you were saying. Probably not, but during the coffee break, everybody has the option to talk to you. In your presentation, you were saying that you have done cytocompatibility testing with Alma Blue, I suppose, if I remember correctly. Any hemocompatibility studies so far already or not?
Using Alma Blue?
Yeah, you did cytocompatibility, yes. Okay?
Yes.
With which method?
Using cell lines.
Cell lines, okay. But the detection method?
I did not.
I think in your abstract when I was reading that you use Alma Blue.
Yes.
Alma Blue. Okay. Have you done already any hemocompatibility studies?
Until now, only compatibility studies using
with cell lines.
Yes.
Okay. Good. Okay. So now we're coming from the sunny Brazil. Okay. We would need some sunshine in Basel today. We come a little bit closer to here.